Have you ever wondered how new medicines get approved? From over-the-counter allergy pills to complex therapies that fight cancer, all of the drugs we use today were made possible because of clinical trials. Clinical trials are controlled medical experiments where the efficacy and safety of a new medical treatment are assessed in humans. We'll explore the different phases of clinical trials and the types of data that are collected in each phase. Let's follow the journey of Drug X, a promising cancer-fighting drug. Before any testing of Drug X in humans can begin, there is a preclinical research phase. In this phase, research and development of the drug are finalized, and the safety of the drug is demonstrated in animals. Then, recruitment of human participants can begin. Researchers work with trial matching services to advertise the trial to the right patients. It's important for clinical researchers to recruit from diverse backgrounds to ensure that their study is as representative of the population as possible. Additionally, informed consent and ethical treatment of participants are both high-priority elements of clinical trials. Clinical trials may consist of one or more phases, each of which is its own clinical trial. This process begins with a Phase 1 clinical trial. Phase 1 trials consist of roughly 20 to 80 participants. Best route of administration, appropriate dosage, and analysis of how the drug affects all systems of the body are studied here. If all goes well in the first phase, Phase 1 trials are followed by Phase 2 trials. Phase 2 trials consist of 100 to 300 participants. Researchers ask similar questions as in Phase 1, but since there are more participants, they can analyze subgroups by factors such as age and sex, and study health outcomes based on those factors. With a patient cohort of this size, researchers can also identify blocking factors such as age or sex when designing the study. They can then stratify the patients by these factors before randomly assigning treatments to ensure even representation across these groups. After the successful completion of Phase 2 clinical trials, researchers can proceed to Phase 3. Phase 3 trials consist of 300 to 3,000 participants. Large-scale efficacy data is used to confirm and expand on the results observed in Phase 2, which involves a larger and more diverse participant pool. Additionally, researchers compare the intervention against existing standard treatments or a placebo. Participants in this phase are randomly assigned to a treatment group. The patients who receive the new drug are in the experimental group. Patients who receive the existing standard treatment are in the positive control group. These patients are expected to improve. This group allows researchers to compare the new treatment to the current standard. A placebo is a type of negative control for clinical trials. It's an inactive version of the treatment and allows researchers to compare the effect of the new drug to a treatment without a biological effect. Clinical trials don't end here. In Phase 4 clinical trials, long-term effects are studied, as well as adverse effects that are rare in the population. Thousands to millions of participants are involved as the drug becomes widely available. There is no enrollment phase. Instead, mechanisms are created to capture data from regular patients who take the new drug. Researchers continue to assess the safety of the treatment and look for rare adverse effects in the general population. They also confirm that the effectiveness of Drug X in the field is consistent with its efficacy in past trial phases. So from over-the-counter allergy pills to complex cancer therapies, clinical trials make our medicines safer and ensure we get the best treatments possible. Want to know more about how Octalsoft's AI-powered eClinical suite can help expedite your next clinical trial? Book a demo with us today. Octalsoft. Technology simplified.